Hello, Jocelyn McCarthy, your Biology 225. Um, Today, I'm going to go ahead and talk about this week's lab experiment. And we did the antibiotic sensitivity Kirby-Bauer diffusion test. So I'm going to go ahead and tilt down the camera to show you my agar plate and see what's going on this experiment. Okay. As you can see, this is the agar plate that was created for this lab report, for this lab experiment. Okay, so the whole purpose of this experiment is to do the sensitivity test by performing the Kirby-Bauer diffusion test. So the this method is actually used in the hospital laboratories to assess bacterial antibiotic resistance. What they usually do is that they, at a hospital setting, a bacteria is placed on an agar plate and testing the concentration gradient. Basically, it assesses how once you spread the active culture right here, just like we did in this lab on this agar plate, by putting different antibiotics and see the concentration gradient of which antibiotic grows the most bacteria versus the least resistance. So as you can see, we have 3 different antibiotics that we were asked to use, and one of them is the penicillin right here, Gent for gentamicin, and novobiacin. So I'm going to talk about the 3 different antibiotics that were used for this experiment right here. So let's start off with the penicillin, because that's the most popular antibiotics that we know. Penicillin is a bactericidal narrow spectrum drug that targets primarily the gram-positive bacteria. So what it does is it inhibits the formation of the peptidoglycan cross-links, and that results for cell death. So that makes sense because amongst 3 of them here, if you notice, penicillin has the least growth of bacteria compared to the other 2. I'm going to talk about the novobiacin now. Novobiacin is a narrow spectrum antibiotic. What it does is that it targets the DNA gyrase, and and the gram-positive bacteria as well. So what is that? Is that it stops the bacterial cells from growing and dividing, and without. So, oh, what did I say? Okay, so the DNA, the novobiacin, the narrow spectrum antibiotics, it targets the DNA gyrase and in some gram-positive bacteria. So, all right, so I'm going to move on to the gentamicin now. Gentamicin is also familiar with me at the hospital, that we use it all the time for when we use the triple antibiotic. Say, for example, today we had a patient that had a possible necrotizing fasciitis. So then the surgeon actually requested for a triple antibiotic, and one of them that we use usually for all cases across the board, not just for necrotizing fasciitis, would be the gentamicin, a combination of gentamicin. What else do we use? Bacitracin and Ancef all together in a saline to irrigate the wound usually, and that goes for all services. Sometimes neuro would use it as well for the back, neurospine, and likely in all wound debridement that uses triple antibiotic, we combine all those 3 to make it stronger, unless of course it's specified that the patient has allergic reaction to certain ones. So I'm going to talk about the gentamicin here. Gentamicin is another narrow spectrum bacteriostatic antibiotic. It targets aerobic gram-negative bacteria. So what it does is that it binds with bacterial ribosome, and it's responsible for protein synthesis. So with the results that I have here with the Kirby-Bauer test, the most growth I would say would be the gentamicin, and next one is the novobiocin, and the least growth would be the penicillin. So it tells me that actually the penicillin is the most effective antibiotics that we can use for any staph infection that we have, and that makes sense because it's widely used. As long as we don't misuse the purpose of this antibiotic, then we can prolong the use of this antibiotic. I'm sorry, I didn't know what to say on that one. I kind of blanked out. So, all right, well, that's all for tonight. I'm going to go ahead and tilt the camera again here. That's all for tonight's lab report, and I'll see you again next week. Thanks for watching. Bye-bye.